Today we will discuss about this perineural invasion in pancreatic cancer. So this is the brief overview how this perineural invasion occurs. That is how the pancreatic cancers spread or migrate through the nerves. So we will start. We will divide this discussion in three different sections that is been represented by three different panels. So we will start with the panel A. So in this panel A. Receptors that are CR4 and CXCR1 will interact with interact with the cytokines that are released by neuron, that is CXCL12 or CXCL1, and these interactions, these cytokines, when interact with the receptors of the pancreatic cancers, it will promote tumor invasions, migrations. And apart from this, the glutamate that are released by neuron will go through these NMDAR receptors, and it will upregulate these CA-MK2 or ERK MAPK pathways uh, through this through these mRNA modifications at 6A, and that will cause mRNA modifications of this HK2 proteins, and that will further promote this PNI, that is perineural invasions. And apart from this, there is upregulations of this CD74 within the pancreatic cancers that will lead to the upregulation of the survival pathways, that is AKT or ERG1 pathways, and that will cause this GDNF cytokine release in the tumor microenvironment, and this also supports or favors the tumor invasions or migrations. And the pancreatic cells also, or the pancreatic cancer cells, PDAC cells, also release this SEMA 3D, and that will interact with this PLX and D1 receptors on the neuron, and these interactions will also promote the tumor cells invasions or migrations of the pancreatic tumor cells to the through this neuron. And PNI, the tumor PNI, that is perineural invasions. It means that tumor cells that are close, pancreatic tumor cells that are close to the neuron. So this is the how they look like. So after understanding the factors that will promote tumor invasions and promotions, we will go through these SIS1 cells, how these SIS1 cells differentiate themselves and into the pancreatic cells, or helps the pancreatic cells towards this PNI, that is perineural invasions. So this SIS1 cells has this characteristic, that is SIS1 cells has the characteristic that it will release this cytokines, that is CCL7, that will interact with the CCR1, and that will cause this STAT2 upregulations, and that will finally lead to this TMP1 upregulation pathways. And this TMP1 upregulation pathway will cause proliferation and migrations of the pancreatic cells towards this SIS1 cells. Along with this, it will also cause the upregulations of this CD63 or survival pathway in the SIS1 cells, and that will accelerate or aggravate the proliferations and migrations of the cells. And apart from this, the SIS1 cells also release this CCL2 that helps in the differentiation of these immature monocytes towards the M2 macrophage, and this M2 macrophage then release this CTSB, and that promotes this that helps in this perineural invasions. And apart from these factors, the other factors is that there is a high productions of this MMP1 within the pancreatic cells that cause this upregulations of this anti-3 TRKC pathways, and this upregulation of these pathways is also helps in these perineural invasions. So these are some of the pathways in the that helps. These are some of the pathways or interactions between SIS1 cells and the pancreatic cells that helps in this perineural invasions. Now, in the third part, we will understand how these pancreatic stellate cells and the pancreatic stellate cells and the inflammatory Echinard cells helps in this PDAC towards perineural invasions. So these pancreatic stellate cells will release this NGF cytokines that will interact with this TRK1 and helps in the upregulations of this or help in the production of this microRNA 21,5P, and this 21,5P microRNAs will in will help in the upregulation of this Warburg effect. And we have discussed this Warburg effect in our previous video, where we have discussed we understand that how this because of this Warburg effect, glycolysis pathways uh, does not require mitochondria, whereas the anaerobic or no no whereas the this aerobic glycolysis pathway, that is glucose converted to the pyruvate through the glycolysis pathways occurs within the cytosols itself. And apart from this NGF, the HGF is also released. HGF cytokines is also released by PSC, pancreatic stellate cells, and that will interact with the c-Met receptors and lead to the upregulation of this mTOR pathway. And this mTOR pathway will further cause this NGF release from the pancreatic cells, and that can interact with this TRK1 or reverse interactions, and that will helps in this pancreatic cancer cells invasions towards neuron. And from this inflammatory acinar cells point of view, it will release these pancreatic associated proteins, or PAP or REG3A, that helps in the upregulation of the or triggering that triggers this GAK or STAT pathway through the phosphorylations. And it also has a role or it and this factor upregulation of this JAK-STAT phosphorylations pathway will enhances these perineural invasions. So these are the brief overview, or what are the factors or signaling pathways that will promote perineural invasions in pancreatic cancers or pancreatic ductal adenocarcinoma. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.